People ask, how long does this process take? Your book shows you in humans. In yeast, if they are in an incredibly happy situation, they can divide themselves about once every 16 hours. The vast majority of that time is in interphase, where you are doing all your normal cell things. You are growing, you are duplicating your DNA. And while you're duplicating your DNA, it turns out you can also do all the normal cell things that are going on. But in any case, most of your time is spent in interphase, and only a very small amount of time is spent actively doing the chromosomal division that is mitosis. You don't need to know the exact details here, just the idea that if you're dividing on the order of once a day, about an hour of that time is spent in mitosis. So more like 5 to 10% of your time can be spent in mitosis, and almost all the 90-ish, 90+% of your time will be spent in interphase. There's a really critical thing we haven't talked about a lot, and that is, how do the cells decide they want to do mitosis, they want to divide? And there's 3 checkpoints that are going to be important. We're going to give brief descriptions of them, and then when we talk about cancer genetics, we're going to look at them much more fully. But between G1 and S, there's a checkpoint known as the G1 or G1S checkpoint. Sometimes biologists ran out of crazy words. And at this point, this is the do I go to S phase point? Do I pause in G1 and wait until I have enough food and I've grown big enough? Or do I decide that I am a cell that never wants to divide again? That sort of cell might be a skin cell. It might be another cell that that's not going to divide, that instead relies on what are called stem cells to make more of them. So skin cells, blood cells, a lot of those cells are never going to divide again, and they go into what's known as G0, which just means they stay as fully functional cells but can never go into a division process. So how does the cell decide which phase it wants to go to? Well, it uses various proteins, and again, we'll look at these later, but it uses various proteins to sense nutrient availability. Is there a lot of basically ATP in the cell, or a lot of AMP, which is the energy poor form of ATP? Are there growth factors being shot at the cell? So for example, if there are no cells touching that cell, well, that would be a good reason you might want to grow. Maybe you have a cut and now you don't have cells touching your skin cells. It's time to grow. Maybe you're a juvenile organism and you need to grow bigger. If your cells or if your chromosomes or your DNA is damaged, you need to fix it before you can copy it, because the last thing you want to do is copy a bunch of mistakes. And there's various DNA stress factor proteins. And the final thing is you need to be big enough you are able to divide in half, and there's ways that the cell can mechanically sense how big it is. The next checkpoint we want to talk about is the G2 checkpoint. This is the point where the cell has to be ready to not have any more transcription and translation occurring for the next hour or so while it goes through mitosis. So it has to be 100% ready for mitosis. It checks specifically for DNA damage and to make sure that chromosome replication is complete. It will also check to make sure that the nutrient availability is still good. Well, what happens if things are bad? It's kind of a cool process. So the cell can either say, if things are bad, I can wait until my DNA has been fixed by DNA repair enzymes, and that my nutrient stress has gone away. So somebody dropped an ice cream cone and now I have plenty of food, or something like that. Or it can say, wow, I'm in terrible shape. It would be better for me to die than to replicate out of control, or to replicate my really bad self if I'm in a multicellular organism, because that could actually poison all the other cells. And that's more or less what cancer is, when the cell doesn't kill itself when the cell starts replicating out of control. And it's got a great name. It's called apoptosis. Probably 90% of people say apoptosis, but I worked for an apoptosis researcher guy and he said apoptosis, so I say apoptosis. If anybody ever questions how you say a scientific term, just say, the first person I heard it from said it this way and I just picked it up as a habit. It's a great excuse. It also means that you may have learned it by reading it, and so you may have a different pronunciation because it's hard to tell what pronunciations are in English. In any case, if the cell decides to go through apoptosis or programmed cell death, you do cool things like membrane blebbing and nuclear membrane collapse, and then the cell basically disintegrates. It's all done by these things called caspases, these proteins called caspases, and we'll talk about them in chapter 19 or something, which is the cancer genetics chapter. If you want to see, this is a very simplified version of some of these pathways, but basically there is a master protein that either says die, or there's signals that break this protein down and then the cell lives. And then there's all these other proteins that either are saying, yes, we're in good shape, so destabilize the p53 and be ready for the cell to continue living, or, oh no, things are really bad, there's stresses all around, stabilize that protein and go to apoptosis. And they've got great names like survivin and things like that. So we'll talk a lot more about these sorts of pathways in chapter 19. You don't need to memorize this by any stretch of the imagination, but I was just getting you a little flavor of why we care about some of these pathways. The last checkpoint is the M, which looks like it's right where the G2 one was, but this is actually at mitosis, within mitosis at metaphase, and that's where the cells, the chromosomes have to be aligned on the metaphase plate, and the cell cycle will pause there until they're all lined up. And it's not that the cell is actually counting all the chromosomes on the plate. The cell is sort of sweeping and making sure there are no chromosomes anywhere except the cell phase, except on the plate. So it doesn't need to be able to count to the 46 chromosomes you have. It just needs to know that everything is on the metaphase plate, not still floating around the cell.